My name is Jonas Karlsson, and I work here at Google. And today we have a very interesting speaker, Aubrey Gray, visiting us for the third time. And he's going to talk about the aging of the avid genome, a decisive but ambitious solution, and some tentative alternatives to this. And he's based in Cambridge, UK, and he's main researchers at Meflusa Foundation. And I think we're going to have a very interesting talk here. Thank you.
Thank you very much, Jonas. Can everybody hear me? Jolly good. Yes, thank you very much for inviting me back. This, as Jonas said, this is the third time that I've spoken at Google, and I must say I enjoy these talks enormously because the audience is, of course, predominantly not biologists, but I know that a lot of you do have at least a certain amount of expertise in this sort of thing. So I'm going to try and, as usual, give a talk which tries to be approachable to non-biologists, but also to have a certain amount of depth to it. One thing I want to mention before I start is that since I was here last, the book that I published, that I wrote over the past couple of years has come out. Jonas has a copy over there. Hold it. I have a few more of these available to give away for some small amount of money. If anyone's interested later on, it's a book written very much for a general audience, but without cutting any corners on the science, really. So I'm certainly hoping that it's the sort of thing that people of this sort of degree of expertise will get a lot out of. And it's available on Amazon as well, of course, if you feel like thinking about making a decision. So what am I going to talk about today? Well, I will try to go through the following sections in the talk. First of all, I'm just going to give a brief recap of the general philosophy behind the approach that I've developed over the past several years for combating aging. And this is an approach called Strategies for Engineered Negligible Senescence, which is a hell of a mouthful, and the acronym of SENSE is rather more memorable. So I'll just give a brief recap of that, and if anyone wants to know a lot more, then either you can ask me afterwards or you can go and have a look at the video of the talk that I gave here in May. Then I'm going to move on to one particular strand of sense, which is the main topic of the talk today. I'm going to explain why it's important for us to worry about the damage that accumulates during life in what I call the other genome in the title of the talk, in other words, the mitochondrial DNA. Then I'm going to look for a while at some of the approaches that various people have looked at for combating the accumulation of mitochondrial mutations simply by eliminating them and restoring the mitochondrial DNA to a less damaged state. And I'm going to give some reasons for pessimism with regard to the possibility of that approach actually being effective in the foreseeable future. Then for the rest of the talk, I'm going to talk about the approach that I think is going to be effective, which, as you will see, is not so much a repair strategy, a direct repair strategy, but rather a strategy that's better called obviation. In other words, we basically let the damage accumulate, but we do some jiggery-pokery to make the damage no longer be damage. In other words, to make it harmless. Okay, so I'm going to start with this. Now, some of you may remember, if you came to my talk in May, that the first thing I did was to describe what aging actually is. And of course, it's pretty difficult to know what to do about a phenomenon if you want to do something about it until you understand at least a little bit about what the nature of the phenomenon actually is. So I want to, first of all, make sure that a few very basic things are understood. The first thing is, we don't have genes for aging. Aging is something that happens by default. Just like the aging of a car or a house or whatever, we don't have mechanisms built into the human body that say that it should actually last less long than it otherwise would if we didn't have those mechanisms. Rather, what we do have is a whole host of anti-aging machinery built into us that allows us to live as long as we do. And the reason we have aging at all is simply because the anti-aging machinery that we have is not comprehensive. So in other words, if evolution had taken the trouble to invent even more elaborate, even more sophisticated and comprehensive anti-aging machinery than what we have already, then we would age even more slowly than we currently do, and we would live even longer. And I think probably the most succinct way that this has yet been described is what I'm saying up here, that aging is a product of evolutionary neglect rather than evolutionary intent. That was the way that a colleague and friend of mine, Len Hayflick, put it some years ago. All right, so that's the first thing you need to appreciate about aging. The second thing is the sort of, if you like, causally two-step nature of aging, that there's an ongoing aspect and there's an eventual aspect. Now, for the purposes of this talk, I'm going to be using the word damage in a manner that is essentially defined by the second and third sentences here. Damage is, for purposes of today, a set of molecular and cellular side effects of metabolism. In other words, side effects of the normal processes that keep us alive and more or less intact from one day to the next and from 1 year to the next. Those metabolic processes incorporate the creation of a whole bunch of damage. As I mentioned a moment ago, most of that damage is automatically repaired by the intrinsic anti-aging machinery that we have built into us, but some of it isn't. And those things that are not repaired accumulate, and eventually some of those things that accumulate start to get in the way of metabolism. They start to cause our metabolism not to work so well, and that's the phenomenon that the set of phenomena that I'm grouping under the word pathology, including all of the age-related diseases that we're so familiar with, and also all of those things that go wrong with us in old age that we would not normally call diseases, things like gradual decline of the efficacy of the immune system, loss of muscle mass, things like that. And finally, of course, as everyone knows, whether you're a biologist or not, it's a downward spiral. It's a sort of vicious circle. When pathology starts getting going, things start to go downhill at an accelerating rate. All right, so that's aging. And as those of you who saw my talk in May or who have seen my work before will be familiar with, this is the essence of the conclusion that I came to several years ago and that has dominated my work in this area ever since. What I'm saying here is that if we accept that there is this chain of events, metabolism causing damage and damage eventually causing pathology, then we can envision three different ways in which we might go about doing something about aging. There's what I'm calling the geriatrics approach, which is to wait until pathology starts to emerge and then to try to hold back the sands of time and stop the pathology from accelerating and accumulating as fast as it normally would. And that's fine in principle. It's a way of postponing the age at which pathology becomes seriously life-threatening and problematic. But, of course, it's a losing battle because the precursors of the pathology, these various types of molecular and cellular damage, are continuing to accumulate throughout life, and therefore the geriatrician's job is becoming harder and harder. Gerontologists tend to start from that knowledge, from the appreciation that the geriatrician's approach is a losing battle, and they say, well, okay, prevention is better than cure. Let's get stuck into this chain of events at an earlier stage. Let's try and clean up metabolism and make it lay down these various types of molecular and cellular damage at a slower rate than it naturally would. That, of course, will have the appropriate effect. It will delay the age at which damage reaches a level that causes pathology to emerge. Problem with the gerontologist approach is that, yes, it's a fine idea in principle, but we just don't understand metabolism well enough. We know a pitifully small amount, really, about how cells work, let alone how organisms work. And the result is that we end up essentially not being able to adopt this approach in a productive manner. If we try to interfere in metabolism, then 90% of the time metabolism just laughs in our face and gets on and does what it was doing anyway. And the other 10% of the time, if we do actually manage to have the effect that we want to have, we turn out that, in general, we'll have a whole bunch of other effects that we hadn't expected because of the aspects of metabolism that we don't understand. So that's why the gerontology approach is an approach whose time has not really yet come. And this led me, way back in about 1999, 2000, to the realization that a third approach might be the way to go. The engineering approach, as I like to call it, I think of as the best of both worlds, because there what I'm saying we should do is let metabolism lay down these various types of damage at the rate that it naturally lays them down, but then let's go in and actually repair that damage ongoingly or periodically so that it doesn't reach the level of abundance that would cause pathology to happen. And the engineering approach has the best of both worlds in the sense that it's sufficiently early on, sufficiently preemptive, that it doesn't suffer from the downward spiral that the geriatrician's approach does. But at the same time, it's late enough in the chain of events that it's not actually interfering in metabolism itself, and therefore it's not attempting to be cleverer than evolution, which is always a dangerous thing to try to be. All right, so I've told you the basic philosophy, and as I say, if you want to know more, then you know where you can find it out. What does this mean in actual practice? What is this damage thing that I've spoken about in the abstract? Well, it's quite easy to see what it is if you just look at what the human body is made of. Here's the body. What are we made of? We're made of cells and stuff between cells. You know, that's a simple breakdown of what we're made of. So what can damage accumulate in, and in what way? Well, let's think about cells. First of all, you can imagine that you could have the wrong number of some type of cell. You could have too many or you could have too few. Secondly, you could have cells where you had the right number of them, but somehow their contents was inappropriate. Either they had stuff in them that you didn't want to have in them, or you had stuff that you do want, but the stuff in question, the molecules inside the cell, have been damaged in some way. So we can imagine that in terms of cell number, it's straightforward. If you've got too many of some type of cell, you just zap them somehow. If you've got too few, you put them back in. And of course, that's what stem cell therapy is about, is basically putting cells back in when the body isn't doing it on its own. In terms of the contents of the cell, you could think of a cell that's got the wrong type of contents as being an example of, a subset of, having too many of a particular type of cell. So you could just say, well, OK, let's zap these cells that have got the wrong contents and replace them with other cells that are OK. But it turns out that that's not always a convenient option, because the cells in question may be rather hard to replace. So it may be actually easier in some cases to repair the cells rather than to replace them. And if they've just got stuff in them that you don't want, you can get rid of that stuff. Indigestible molecules, garbage, basically. If alternatively there's things in the cell that you do want, but those molecules have been damaged, then you've got to do something else. Ideally, you'd like to be able to repair the changes to reverse the changes that have happened to these molecules in the cell. Maybe, however, you won't need to. Maybe the molecules have undergone changes that are not actually pathogenic, or at least not within what we think of as a currently normal lifetime. And there are, in fact, a few examples of changes to the contents of the cell which seem to fall into that category, where we just don't need to worry about them because the change happens too slowly. It doesn't actually cause any problem for metabolism. So those are two options, but there's a third option, which is going to be rather important for this talk, which is what I'm calling obviation. The idea here is simply that, OK, the damage happens, and it's bad for us. It causes pathology in the end. The idea is to make some little tweak to the role of this particular molecule in the cell such that the type of change that's happening to it suddenly becomes a harmless change, when previously it would be a harmful one. And that sounds a little bit like hand-wavy, but you'll see that that's actually a genuinely real option in real circumstances, as I'll come to shortly. Oh, and then finally, of course, when we talk about stuff between cells, that's basically the same deal as stuff inside cells. Either you get rid of it if it's not wanted, or you try and repair it if it's damaged. Same deal, really. OK, so that's basically the essence of how I look at how to go about combating aging. And I'm going to talk now about the mitochondrial DNA. And I'm going to actually go back one slide and explain why. I'm going to do that. Yeah, so mitochondrial DNA is DNA inside the mitochondrion. The mitochondrion is inside the cell. So in other words, it falls into this category down here. Damage that happens to the mitochondrial DNA is damage to something that's inside the cell, and we don't want to get rid of the mitochondrial DNA. We need it. We want to fix it if we can. But there are these various options, as I said. Can we ignore it? I'm going to be answering that question in a moment. Can we repair it? I'm going to be having a look at some options for that. And then can we obviate it? All right, so first of all, can we ignore it? And the answer is going to be no. Here's why. What damage do we need to repair? Damage that we need to repair, in other words, damage that we can't afford to ignore, is damage that contributes to pathology in a normal lifetime, and, of course, is not repaired automatically by our existing inbuilt anti-aging processes. Now, another way of answering this question is that the damage you need to repair is damage that happens to long-lived molecules, and in particular molecules whose function in the cell is essential. If we think about a short-lived molecule, let's say a protein. A protein is constructed in the cell, and it has some job to do, and it does the job for a while, and then it gets damaged in some way. Let's say it gets oxidized in some way. All right, so what then happens? Well, in general, what happens is the protein will simply be broken down. And the undamaged amino acids that the protein contained will be reused in some other proteins, and the damaged ones will be excreted. Now, that means that the damage isn't accumulating. The damage is gone. So that cannot count as damage by the very narrow, strict definition that I'm using for this talk. The only molecules in which damage actually matters are molecules that are not broken down and recycled, but molecules that live for the lifetime of the cell. And the obvious example, of course, is DNA. In fact, really, we can say that there are only two types of molecule that qualify as damage in this way. One is DNA, and the other is indigestible molecules. I mentioned indigestible molecules earlier on. Things that the cell creates, usually by accident, but then when it wants to break them down, it finds that it hasn't got the enzymes to do it. Somehow, for whatever reason, the molecule is of a type that the machinery that it has to break things down doesn't work on. And sometimes, for various reasons, cells will not want to excrete the molecules in question into the environment either. So molecules like this can accumulate inside the cell, and those things cause a whole bunch of other problems. If you want to know more about that, then read chapter 9 of the book or come to my next talk, which hasn't been scheduled yet, you understand. But anyway, so that's one thing that goes wrong, but the DNA is the main thing that accumulates damage that we have to do something about. And as I mentioned, there are two genomes in our cells. One is the genome that we're all very familiar with, that consists of 23 pairs of chromosomes and a very large number of base pairs. And the other is the mitochondrial DNA, which is present in between a few hundred and a few thousand copies, depending on the type of cell, in nearly all of our cells, and which is much, much smaller. Those of you who are sitting near the front will be able to see here the number 16,569. That is the number of base pairs in the mitochondrial DNA of humans. So in other words, it's an absolutely minute fraction of the size of the nuclear genome. And indeed, there are only 13 proteins encoded in the mitochondrial DNA. This is a gene map of the mitochondrial DNA. Everything except this little bit up at the top here encodes something or other. These two little things over here encode RNA molecules that do not encode proteins, and all the other ones encode proteins, except for the few little tiny things in here which are transfer RNA genes. So it's very, very small indeed. And that turns out to be quite convenient, as you'll see. So I mentioned, first of all, can we actually ignore damage to the mitochondrial DNA, or do we have to do something about it? Well, the first thing we can say is that, unfortunately, it does accumulate damage. It's pretty easy to demonstrate that older people have more mutations in their mitochondrial DNA than younger people. Does the damage matter? Well, I have to be honest with you here and tell you that at the moment, biologists do not know for absolutely sure and certain whether or not mitochondrial mutations actually matter in a currently normal lifetime. But it looks as though they probably do. There's lots of correlative evidence, lots of circumstantial evidence that suggests that indeed these things do matter. And that's bad enough to be interesting. I take the view that it's not appropriate to spend too much of our time gazing into our navels and trying to work out whether such and such a phenomenon actually matters in aging or not. It's actually better to go out and damn well address the problem, even if we don't know that it's a problem. If we end up fixing a few things that we find out in the end we didn't need to fix, no real harm done, whereas if we spend too long not doing anything because we're too worried about whether something matters or not, then we may actually delay progress. So this is the sort of evidence that exists that shows that mitochondrial mutations may seriously matter. And what this basically shows is that mitochondrial mutations are not distributed evenly across tissue. What's been done in this slide is someone's taken a chunk of skeletal muscle and sliced it perpendicular to the muscle fibers, so that each of these circular things is a cross-section through a muscle fiber. And then they've stained it for the activity of an enzyme called cytochrome C oxidase, which is one of the enzymes that some of whose subunits are encoded by the mitochondrial DNA. So if the mitochondrial DNA is damaged, then the cytochrome C oxidase enzyme is not going to be constructed. Now, what we see here is that there are entire muscle fibers, well, muscle fiber segments anyway, in which there is no cytochrome C oxidase activity. I told you a moment ago that there are hundreds of copies of the mitochondrial DNA in each cell. In a muscle fiber, there are thousands per millimeter of the length of the muscle fiber. And they've all, or more or less all anyway, lost the ability to make this enzyme. Or otherwise, there would be activity being shown here for this enzyme in these particular muscle fibers, and there aren't. So what we've actually got here is somehow cells or muscle fiber segments being entirely taken over by mutant mitochondrial DNA, whereas the neighboring cell is perfectly fine and does not have a detectable deficiency of mitochondrial DNA. So that's a pretty interesting thing. It allows the possibility that these cells, even though they are relatively rare, even in old people, are nevertheless toxic, are in some way poisoning the rest of the body. And a lot of my work back in the late 90s was actually focused on the detailed mechanisms for that. I'm not going to talk about that today. Now, I said earlier on that when there is a molecule in a cell that's damaged, we have a couple of options if we decide that we have to address the damage. One is to repair the damage and actually restore the molecule to the structure it had in an earlier age, and the other is to obviate the problem. So I'm going to look at repair first of all. Now, there are various options for how to go about repairing the mitochondrial DNA. The most obvious one, of course, is just to take new copies of the mitochondrial DNA that are not damaged, that have the normal unmutant sequence, and introduce them in large numbers into the body, especially into the cells that have been affected, have been taken over by mutant mitochondrial DNA. And that sounds like a damn good idea. It sounds pretty tricky. You know, getting molecules that are 16,000 base pairs long into cells in the body is a lot more ambitious than most gene therapy. But various techniques have been tried over the past 10 or 12 years, and actually, at this point, it would be premature to dismiss out of hand the possibility that we could do this. So this is something that we might usefully think about. But there's a big, big problem with using this to tackle the phenomenon of mitochondrial DNA mutation accumulation. And the problem is that the reason we have this punctate distribution, where some cells are accumulating mitochondrial mutations and others are not, is because when a mutation happens in one copy of the mitochondrial DNA in a cell, it seems, at least sometimes, that that copy somehow gets a selective advantage. It gets replicated more often than the rest of the mitochondrial DNA. And you have a phenomenon which is called clonal expansion of the mutant mitochondrial DNA at the expense of the non-mutant. I've got a picture of how we found those sort of things out. Those of you who know a bit of biology will recognize this as something called a southern blot, which is a way of determining whether, essentially a way of determining the length of a particular chunk of DNA. And what this is showing, to cut a long story short, is that if you take a whole number of muscle fiber segments and you look at the DNA that's in them, this one on the left is a control for measurement purposes. If you look at the amount of DNA, mitochondrial DNA in these things, and you ask what length is it, then most of these cells have all the same length of this particular segment of the mitochondrial DNA, showing that essentially if they've got any changes to the mitochondrial DNA, then the changes are not changing the length. They will be point mutations of one sort or another. Whereas these three have got different lengths for that particular chunk of DNA, indicating that there's some DNA missing. And as you can see, there's just one particular length in this cell. There's none of the normal length. There's also none of any other length. There's not a smear there showing a range of different lengths. There's just one length, and a different length in a different cell. This is the sort of thing that was discovered way back about a decade ago and discovered to be absolutely universal in this, that you get this clonal expansion of single mutations. Now, that's really bad news for the idea of putting back new copies of the normal mitochondrial DNA, because if you put them back, yes, the cell that you put them into may be happy for a short while, but only a short while, because the wild type, the non-mutant mitochondrial DNA that you have introduced, will go the same way that the original mitochondrial DNA did. It will be eliminated in favor of the wild type of the mutant mitochondrial DNA. And the exact mechanism for this clonal expansion is still a matter of debate, even though people have put forward a large number of theories of this over the years. But it really doesn't matter what the mechanism is. The fact is, it happens, and therefore replacing the mitochondrial DNA seems to be a bit of a non-starter. Well, here's another possibility. OK, there's this clonal expansion going on. Why don't we try and superpose, shall we say, on that phenomenon, a reverse phenomenon? In other words, why don't we just make it really hard for the mitochondrial DNA that's mutant to actually be replicated, so that essentially we impose a selective advantage for the non-mutant mitochondrial DNA that would outpace, so to speak, the natural selective advantage that the mutant DNA experiences. And people have been trying to do that, actually. A group from Newcastle have been really leading the way. They found some very weird analogues of DNA that are able to bind to DNA in a way that stops it from being replicated. And these molecules are sequence-specific. So you can actually introduce these molecules into cells that have a particular mutation, and the mutant mitochondrial DNA won't be replicated, and the wild type, any residual wild type mitochondrial DNA that's left will replicate instead, and the cell will be restored to some sort of mitochondrial health. And that's a pretty good idea in principle, but there are a couple of things wrong with it as well. First of all, I've shown you already that even though in each individual cell there is a clonal expansion of just one particular mutant sequence, nevertheless in the next cell that's affected, there'll be a different mutation that's clonally expanded. So in other words, a single DNA analog that's stopping the replication of a single sequence will not be very much use in terms of combating the accumulation of mutant mitochondrial DNA in aging. There's also the second problem, which I could go into details about later if you want, but basically that it's possible that you'll end up making cells that have no mitochondrial DNA at all, whether it be mutant or non-mutant, and that's just as bad as having entirely mutant mitochondrial DNA. So anyway, the long and short of it is this really doesn't work. However, it turns out that one shouldn't give up. A couple of years ago, some friends of mine working in Virginia came up with a very creative idea, which is a good deal more promising, which is essentially to combine the two ideas that I've just told you are hopeless into an idea that's not quite so hopeless. The idea here is we again introduce mitochondrial DNA that's working, that does encode all the 13 proteins it's supposed to, but we mess around with it in such a way that it destroys the DNA that it comes across that's there naturally. The method that they've used to make this happen is by introducing into the mitochondrial DNA something called a restriction enzyme gene, such that the DNA doesn't only encode the 13 proteins it normally would, it also makes this thing called a restriction enzyme, which is something that cuts DNA. And it cuts it at a specific sequence. And the trick that was used was to use a restriction enzyme that cuts a sequence that actually does occur in normal mitochondrial DNA, preferably a couple of times, so that both normal mitochondrial DNA and more or less any mutant version of mitochondrial DNA will be destroyed, will be cut up by this restriction enzyme that the new DNA encodes. And the way that the thing has been made to work is by altering the engineered mitochondrial DNA so that not only does it encode this restriction enzyme, but also the places where it would normally be cut by that restriction enzyme have been modified so that it's no longer a substrate for the enzyme, so that the enzyme doesn't cut it anymore. So this is a sort of invasive mitochondrial DNA molecule. It's an incredibly clever idea, and the most amazing thing is that they've more or less got it working, but it's all still rather preliminary. Most of this work hasn't been published yet, so I can't tell you all that much about it at this point. However, this is the name they've given to it, protofection, so if you come across that in the papers any time, then you'll know what it's all about. Another approach to all of this that shouldn't be dismissed out of hand is something that I alluded to towards the beginning of the talk, which was that we could actually replace the cells. We could actually zap the cells that are affected by mitochondrial mutations and put new ones back. And that's not completely ridiculous, but unfortunately there are some problems with it. The first problem is that the cells that are mainly affected by this are cells that are actually not very easy to replace. Muscle fibers I've told you about, also neurons in the brain. Maybe we could use stem cell therapies to replace this, but it's a bit of a tall order, and it's a particularly tall order for skeletal muscle, because it turns out that in skeletal muscle, if you try to kill just a small segment of a fiber that's been overtaken by mitochondrial mutations, for example, then you're very likely to kill the whole fiber because of separating it from its motor neuron, and that's something that will probably do more harm than good. So I don't have very much optimism for that approach either. All right, so what I've told you so far is that there are a whole variety of different ways that we might go about combating the mitochondrial DNA mutation problem by repairing it. And they all look, well, maybe some of them might work, maybe some of them won't, but it's all a little bit iffy. It would be nice if we had an approach that was a little cleaner. And we might have one. And that's what I'm going to spend the rest of this talk discussing. It's an obviation approach. In other words, what it's going to be about is let mitochondrial mutations accumulate, don't try and repair them, just make them harmless. So how do we do this? Well, first of all, we have to ask, what does the mitochondrial DNA encode? I told you it encodes 13 proteins. I haven't told you what those proteins do. What they do is they are part of the machinery that does the chemistry of breathing. That is, oxidative phosphorylation, the process that combines electrons from nutrients like carbohydrates and fats, combines those electrons with oxygen to make essentially water, and in doing so, it extract energy from those nutrients and makes ATP, which is, of course, the universal currency of energy within the cell that allows us to do all the other things we do, like protein synthesis and DNA replication and whatever. Now, OXPHOS is something that we're not going to end up being able to do without. We need OXPHOS. Because, if we couldn't make ATP in the amounts that OXPHOS does, then we basically wouldn't be able to do most of the things we do. So we could, in principle, make a sort of substitute process that did what oxidative phosphorylation does, but that is an unimaginably tall order. Oxidative phosphorylation has only been invented once in the whole of evolution. It's a very elaborate system. We ain't going to reinvent it. One possibility is that we could look at some of the components of oxidative phosphorylation, the components that are encoded by the mitochondrial DNA, and in some way create substitutes for them. And there are one or two options along those lines that, well, I haven't got time to discuss now, but that involve essentially going and looking at what other species do. Some other species have found ways to do without certain components of the oxidative phosphorylation machinery, and it's been speculated anyway that we might be able to exploit those tricks. But we're probably not going to be able to solve the whole problem that way. So the approach that I want to look at in more detail is the bottom one here. The idea that, yes, we need OXPHOS. Yes, we need to encode to create OXPHOS using the same machinery as usual. But let's make that machinery, let's introduce that machinery in some other way that does not rely on the mitochondrial DNA. And that turns out not to be completely implausible. So I mentioned already that the mitochondrial genome is very small, that it's only about 16.5 kilobases long, and that it only encodes 13 proteins. What I haven't mentioned yet is that the mitochondrion itself is a really, really complicated machine, in which there are at least 1,000 different proteins. All the others are already encoded in genes in the nucleus of our cells, in our normal genome. So this is a graphical summary of how mitochondria are constructed. You've got our normal genome up here, and you've got our mitochondrial genome here. The mitochondrial genome encodes 13 proteins, and the nuclear genome, apart from all the non-mitochondrial proteins that it obviously also encodes, it encodes maybe 1,000 or so proteins that are part of the mitochondrion. There's a machinery in the membrane, in the surface of the mitochondrion, which imports those proteins that are encoded in the nuclear genome into the mitochondrion, so that they're in the right place, just as if they had been encoded in the mitochondrial genome in the first place. And then there's a whole bunch of complicated process of assembly of those proteins into various complexes and so on to make the extremely intricate machine that we call the mitochondrion. So mitochondrial biogenesis comes from two different genomes. Now, here are some convenient facts. The first thing I've just mentioned to you is that something like 99% of mitochondrial proteins are encoded in the nucleus, not in the mitochondrial DNA. Second thing is that that's actually true for OXPHOS as well. The oxidative phosphorylation machinery in the mitochondrion is only one of the things that mitochondria do, but it consists of perhaps 100-odd, something like 80 proteins. So something in the region of 80% of the proteins that are parts of the OXPHOS machinery are already nuclear coded. Now, here is the really useful fact, the really convenient fact. The nuclear DNA does not accumulate mutations anything like as fast as the mitochondrial DNA. Now, exactly why that's the case is a matter of some speculation. I think that it's got a great deal to do with the fact that mutations in the nuclear genome can give us cancer, even if they affect just one cell, and they can kill us that way, whereas mutations in the mitochondrial genome can't give us cancer, and so therefore they can only kill us if quite a few cells get affected. And the talk I gave here in June was an elaboration of my views on cancer and how we are going to go about it. That also is, of course, online, if anyone wants to know more about it. But anyway, the fact is that the nuclear DNA is much, much better protected. It accumulates mutations much more slowly. I've told you that there's this machinery in the surface of the mitochondrion that imports proteins from the outside onto the inside of the mitochondrion so that they can be assembled into the mitochondrial structure. And we understand the machinery, the protein import machinery, pretty well. It tends to be called the Tim-Tom complex, believe it or not, translocase of the inner mitochondrial membrane, translocase of the outer mitochondrial membrane. And another useful thing that I'll come back to in a minute is that nearly all the mutations that we actually see in the mitochondrial DNA in normal aging are mutations of a type that knock out all 13 proteins. So you don't have mutations that cause the construction of bad proteins. It's either good proteins or no proteins. That's going to be a very useful fact. So what does all that add up to in terms of a proposal? Well, this is what it adds up to. And this is a proposal that was first put forward back in the mid-1980s, actually, and it's called allotopic expression. The idea is that we take these 13 genes from the mitochondrial DNA and we modify them in various ways so that the proteins that they encode, if those proteins found themselves on the outside of the mitochondrion, would be imported into the inside, just like the 1,000 proteins that are naturally imported in that way. Having made those modifications to these 13 genes, we put the modified engineered genes into the nuclear genome using standard methods, gene therapy, transfection, whatever, depending on what you're doing. Now, what's the effect of that going to be? Well, the first effect is that these genes, these proteins that the genes are creating, will be harmless to healthy mitochondria. You may think, well, maybe there'll be too much of some proteins in the mitochondrion. Turns out that the mitochondrion is really good at coping with flexibility on that, so that's not a problem. Secondly, once a mitochondrion does end up being mutant, it won't matter anymore, because the proteins that the mitochondrion is no longer making on its own, because its mitochondrial DNA is no longer making proteins, those proteins will be arriving from the outside of the mitochondrion, and so the mitochondrion will be able to assemble its oxidative phosphorylation machinery just the same way that it ever was able to when it had the proteins. And here's the real kicker. There's a hell of a lot that we don't know about the detailed way in which the various oxidative phosphorylation enzyme complexes work and how they're assembled and all that sort of thing, but we don't need to know any of that in order for this approach to work. Because what we're doing is we're putting the same proteins into the same place that they naturally are, so that the machinery for assembling them into complexes and making them function in the normal way can be absolutely unaltered. And because all that machinery is already encoded in the nucleus, that none of that machinery is going to be broken in a mutant mitochondrion. So it all just ought to work, irrespective of the things we don't know. This is, in other words, a classic case of an engineer's approach to anything, really, sidestepping our ignorance, sidestepping the things that we don't know. So this is essentially what it comes down to, going back to the picture I showed you before. This is the standard situation, and rather than relying on the mitochondrial DNA to make these 13 proteins, we take the genes, we do some molecular biology to them, we stick the modified genes back into the nucleus so that a few more proteins are constructed in the cytosol, in the inside of the cell, and imported into the inside of the mitochondrion so that they work normally. A lot of people have been skeptical about this proposal over the years, including before I even got involved in this. But all of the proposals that have been put forward for why this won't work have been shown to be flawed. In fact, a lot of them didn't even need to be shown to be flawed. They were just obviously flawed. Things like, for example, the mitochondrion uses a different genetic code. So certain codons, certain triplets of nucleotides mean a different thing in the mitochondrion than they do in the nuclear DNA. That's something that's trivial for us as molecular biologists to alter, to revert in the process of making our engineered genes. So it's something that evolution would find to be a big barrier in spontaneously, naturally moving these genes to the nucleus, but we don't have a problem with it at all. Some people have said that maybe if these proteins were constructed in the outside of the mitochondrion, in the main body of the cell, then they would be toxic in some way. But actually, people have been looking pretty hard for evidence of that, and they haven't found any. There's some toxicity if you make much too much of the protein, so that it aggregates and generally gets in the way of stuff. But if you make a sensible amount, no one's found any toxicity at all. Some people said, well, actually, maybe the problem is that the machinery that targets proteins to the mitochondrion doesn't actually work perfectly on these particular proteins, and it will mistarget them. Especially, people were worried that it would target them to the cell surface. Again, that seems to be completely not true. People have, as I said, they've been worried about the possibility of having too much of some proteins relative to others. And lots of experiments have shown that actually the mitochondrion is really good at coping with a wide variation in the relative abundance of different proteins and getting rid of the ones that are present in excess and so on. So that's no problem. And then finally, basically some botanists in general who were looking at chloroplasts and extrapolating from them have put forward a proposal, which I can't actually remember what this stands for, but essentially it's an alternative reason why evolution hasn't done the job for us and put these genes into the nucleus already. And it's a completely broken proposal. So in the last part of the talk, I wanted to discuss how far we've actually got with this. So far, it's all a nice, trendy idea, but the question you'd probably like to know is it going anywhere? And I mentioned that the idea was first put forward 20 years ago, so you may be thinking, well, it can't be that easy, can it? And you're right, it's not that easy. But progress was made early on. In fact, the main reason why people got interested in it was because when this was first attempted, the first gene out of the 13 that people tried it on, they got it working first time in like only a couple of years. This group from Australia took one gene, which happens to be a very small one, and that turned out to be rather important, and they put it into the nucleus, and they were able to show that it was really working. They created a strain of yeast in which the mitochondrial copy of this gene was completely broken, and all the other 12 genes were working okay, and they showed that this nuclear relocated version of this one gene was able to rescue oxidative phosphorylation in this yeast. They tried it with another gene that was a bit bigger, and they couldn't get it to work. That was a few years later. And that was the beginning of a period of extraordinary inactivity in this area, where basically people couldn't get any funding for it because everyone thought it was far too hard, and people essentially got discouraged. There was one isolated breakthrough a few years ago. After a very long time, a group in Bethesda succeeded in getting a bigger gene to work, and furthermore it was in mammalian cells, so it was very interesting, but that hasn't yet been reproduced. And a couple of other papers that came out around the same time from a group in New York and a group in Florida, and which also looked fairly promising, have now been shown to have been basically misinterpreting their data. They thought they were seeing successful function of these genes in mammalian cells when in fact they weren't. So it all didn't look terribly good. But that one isolated experiment from Bethesda was something that was really very promising indeed. It was very direct evidence that it was really working. This is the key figure from the paper that they published, in which essentially what they were able to do was to show that whereas normal cells whose mitochondrial DNA is functioning perfectly are able to grow like this until they get up to a level of density of the cells in culture where something called contact inhibition happens and the cells stop growing because they're too close to each other. So that's a standard curve for healthy cells that you're seeing there, on a log scale you'll note. You've got these cells here, the ones with the open circles, which are cells whose mitochondrial gene is not working and which grow for a little while but eventually just give up because they can't make any ATP. And then the important one is this one here with the open triangles, where they take a little while to get going, but once they do, they grow nearly as fast as the cells that have their mitochondrial DNA working. These, of course, are the cells that have their mitochondrial DNA mutant, but that have the nuclear version of the thing. So this was a very robust rescue of mitochondrial function from this relocated gene. That was pretty good news. So then, well, what's the problem? Why has it taken 20 years to get anywhere, and why is progress still so patchy? There's a one-word answer, which is this: hydrophobicity. It turns out that the process of getting proteins from the outside of a mitochondrion to the inside is non-trivial. It needs to be done in a manner that preserves the integrity of the mitochondrial membranes. If the mitochondrial membrane is punctured, then it collapses the electrochemical gradient across the membrane. And that electrochemical gradient is an intrinsic component of the way that mitochondria work in the first place, how they actually create ATP from nutrients. But it's also a critical component of the way that the machinery works for getting the proteins across at all. So basically, the machinery that gets proteins across the membrane has to get this enormous molecule — a protein is obviously a seriously large molecule — has to get it across without letting protons across. charge across. And protons, of course, are pretty tricky to hold back if you've got a big hole in your membrane. So it turns out that the way that the machinery does this is by unfolding the protein completely. And that's something that hydrophobic proteins, proteins that have a lot of amino acids that really like not to be next to water, hydrophobic proteins just don't like to do that. They like to fold up into a globular state, minimize their surface exposed to water, and they just refuse to unfold. So it turns out that all 13 of the proteins that are encoded in the mitochondrial DNA are like that. They're really hard to unfold. And sometimes you can just by sheer force get the thing to go, and that's why that one tiny protein in yeast was successfully relocated in the mid-80s. But this is, in general, a problem that's very hard indeed. So the question is, well, has evolution given us any clues? And yes, the answer is that it has. The first thing that's rather good news to know is that there is a certain amount of variation of hydrophobicity in the normal human population. It's important for proteins that need to be imported into the mitochondrion not to be too hydrophobic, or the machinery won't work, as I mentioned. But it doesn't matter how hydrophobic the proteins are that are naturally encoded in the mitochondrial DNA. And so, of course, what's happened is that they are, if you like, unnecessarily hydrophobic. And minor changes reducing the hydrophobicity can be made without actually affecting the function of the protein. That's shown quite graphically here. So the blue line is what's called the hydropathy plot, the degree of hydrophobicity along the length of one of these 13 proteins. So the peaks, the high parts of the thing, are the most hydrophobic amino acids, the most hydrophobic regions of the protein. And as you can see, there are a few peaks which correspond to the most hydrophobic parts of the protein. Now, there's a type of algae called Chlamydomonas reinhardtii, which happens to have succeeded in transferring this particular gene to the nucleus. It's the only species that has, more or less. And as you can see, the purple line, which is the hydropathy plot for this protein in that species, is a good deal lower. The hydrophobicity is lower in all of the places where it was particularly high originally. But if we look at the wide variety of different sequences of human mitochondrial DNA that have been put into the database over the past 20 or 30 years, we find that actually there is quite a lot of variation. So this red line is a sort of artificial human mitochondrial sequence constructed using the least hydrophobic amino acids across the whole protein. And as you can see, especially at this beginning of the protein, if you take the least hydrophobic amino acids everywhere, you can get to a situation that's pretty much the same hydrophobicity as the Chlamydomonas mitochondrial gene, much less hydrophobic than the classical sequence. Unfortunately, you can see that elsewhere along the protein it's not so good. In this point, for example, you could only reduce the hydrophobicity a little bit, not nearly as much as you could in the Chlamydomonas sequence. But maybe we don't have to reduce the hydrophobicity by that much, or at least not everywhere, so this could be progress. Furthermore, as I mentioned, this is only human polymorphisms, things that are actually seen in natural human populations. It may be that if we allow ourselves to go a little bit further out phylogenetically, looking at other mammals, for example, it'll still be okay. We'll still get sequences that still function, that still do the job, but we may be able to find more reductions of the hydrophobicity. And indeed, that's what was found. Here are the human sequences in yellow, and the Chlamydomonas sequence is again in purple. The black sequence is the best you can do using any mammalian polymorphism. And as you can see here, in this peak, for example, we're still more hydrophobic than the Chlamydomonas sequence, but much closer to it than we were in the previous slide. All right, so that's pretty good news. That's certainly a way to go. We could just create versions of these genes that were somewhat less hydrophobic than normal, and we would have a respectable chance of being able to do that without compromising the function of the genes. But it turns out that there is an even more promising approach. There are various times in which it seems that the hydrophobicity of a protein doesn't matter as much as as it ought to, so to speak. First of all, in chloroplasts. Chloroplasts are organelles, of course, in plant cells, which are very like mitochondria. They have their own DNA. They have a lot of proteins that are encoded in the nucleus as well. And a lot of people have said, well, that they provide counterexamples to this whole hypothesis for why mitochondrial genes are actually still in existence. But there are good reasons why that doesn't actually count. Essentially, their topology is different. They do things with electrochemical gradients that are on membranes that do not surround the chloroplast DNA. So this really is an irrelevance, even though it's been pointed to a lot. There are some genes that we have that are very hydrophobic, but are nuclear-coded anyway. And it turns out that they've invented a completely different way of being imported, using a different version of the TIM-TOM complex, which is called TIM22. And some people have thought, well, maybe we could use that as a way of getting these proteins across. It turns out there are reasons why that's probably not going to work. But the biggest and most interesting thing about which proteins get across successfully is that when they've only got one big hydrophobic segment, then that doesn't seem to stop them getting across. It's only if the protein has lots of hydrophobic segments that there seems to be a problem. And that's pretty strange, because the process of import is something that, happens, the thing gets pulled through as a single amino acid chain. You wouldn't think that having more than one transmembrane domain would make a difference. And we've now found out what's going on. And it all comes down to a bizarre phenomenon called cotranslational import, which was discovered even back before anyone started thinking about allotopic expression, 35 or so years ago. We still really don't know how it works. In fact, we don't know the basics of how it works. It turns out that it's really rather common, and it looks as though it might be the big solution for importing things, even if they have lots of transmembrane domains. All right, so what is cotranslational import? Well, as you all know, when a protein is made, the first thing that happens is that the DNA is transcribed into RNA, messenger RNA. And then that RNA is translated by the ribosome into a protein. Now, what I've told you so far is that a lot, in fact 99% of the proteins in a mitochondrion are encoded in the mitochondrial DNA, and then the protein is targeted to the mitochondrion and dragged through the mitochondrial membrane. And for a long time, it was presumed that that was the whole story, that the messenger RNA was constructed in the nucleus, like all messages are, was exported into the cytosol, and cytosolic ribosomes would attach to it and make the protein, and then the protein would be translocated to the mitochondrion. And that's the scenario that's being shown on the left here, where you've got these things, these are ribosomes, tracking along this thread here, which is the messenger RNA, and the protein is being created. Each of these is an amino acid, being created gradually. Eventually, the protein is constructed completely. Here it is, and it's targeted to the mitochondrion and pulled through the TIM-TOM complex. Simple. Turns out that for a lot of proteins that are nuclear-coded but mitochondrially localized, what actually happens is that the messenger RNA itself is targeted to the mitochondrion. It's not imported. It's just targeted to the outer surface of the mitochondrion, and it sits there, and the ribosomes sit there, constructing the protein on the surface of the mitochondrion. Now, the big deal about that is that the proteins can then be imported into the mitochondrion while they are still being constructed, while they're still being synthesized. And that, it seems, is a massive boost to the importability of hydrophobic proteins. Because even hydrophobic proteins, they take a little while to fold up into this globular form that is unimportable. So if they're being imported immediately, if there's only this very short sequence of amino acids on the outside of the mitochondrion that has been constructed but hasn't yet been imported, then essentially the hydrophobicity problem is sidestepped. The protein gets in however hydrophobic it is, and it does its folding up on the inside of the mitochondrion, but that's fine, because that's where the cell is already equipped to cope with the globular proteins. So that's the concept of cotranslational import. And here was the big discovery that happened just a couple of years ago, made by a group in Paris that the Methuselah Foundation, my foundation, is now funding. It turns out that as I said, we don't know how this cotranslational concept works, but it turns out that the trigger for it, the thing that determines whether a particular messenger RNA will be targeted to the outside of mitochondria or not, is encoded in part of that messenger RNA itself, and in particular, it's encoded in a part of the message that does not encode the protein. The messenger RNA tends to have bits on the beginning and end that are called untranslated regions, because that's what they are. The messenger RNA is just a sequence of nucleotides but there is a point on that nucleotide sequence which says, right, this is the beginning of the protein. That's where the ribosome starts doing its stuff and synthesizing the amino acid sequence. And then towards the end of the messenger RNA molecule, there's a point that says, right, this is the end of the protein. But there are some more nucleotides after that as well. And the part at the beginning is called the 5' untranslated region. The part at the end is called the 3' untranslated region. And the 3' untranslated region turns out to be the part that matters most and essentially determines whether the message will be targeted to the mitochondrion. So what people have succeeded in doing is taking the protein, the genes of interest, the genes that in the mitochondrial DNA that encode these very hydrophobic proteins. They've stitched onto the end of these genes the sequence for these untranslated regions from naturally nuclear genes that are found to localize their RNAs really, really thoroughly, really faithfully to the mitochondrion. And it works. The proteins are synthesized on the outside of the mitochondrion. And it really works functionally as well in terms of import. This is a graph very similar to the graph I showed you about 10 minutes ago, but it's even more robust. Essentially, we have here again the black line are controls where the cells have perfectly functioning mitochondrial DNA. The line down at the bottom is one where the mitochondrial DNA is not working, or at least not working well enough, and there is nothing in the cell. There is no nuclear version of the gene in question. And then you've finally got this one here, which is the cell where the mitochondrial DNA is not working, but one of the genes has been replaced by a version in the nucleus, and it's been given this appropriate 3' untranslated region so that the protein is synthesized on the outside of the mitochondria, and sure enough, the cell is really happy for even longer than the previous experiment that I showed you earlier. So that's the situation that we're in at the moment. We seem to have more or less cracked this problem, at least in vitro. It looks as though mitochondrial mutations, which certainly accumulate during aging, probably matter as a contributor to the pathology of aging. It looks as though we may well be able not just to repair those things, but actually to make them harmless by putting copies of the mitochondrial DNA into the nucleus, therefore making the mitochondrial DNA superfluous and making mutations in it ineffective in terms of loss of function. Progress in all of this has been very slow for most of the history of the idea, most of the 20 years that people have been looking at it, but progress seems to be very fast at the moment. So watch this space. There will be a lot of news in this area over the next couple of years, and I'm very pleased that the Methuselah Foundation has made a lot of contributions to making that happen. And I'm also very pleased that you came along to listen to the talk. Thank you very much. Happy to answer questions, of course. Cowards. All right. Go on.
So, I haven't finished your book, but I read this section. Has any progress been made on, I'm aware of this, and I think this is great. Has any progress been made on attempting to target the evolutionary program? I know you're saying it's not sufficient, but to reduce the evolutionary advantage that these mutant mitochondria have. Like, do we have any approaches for that?
Okay, so the group that, so the question was, have we made any progress recently in altering the selective advantage-disadvantage of mutant mitochondrial DNA relative to non-mutant mitochondrial DNA? And yes, there has been some recent progress in that area. The group that really pioneered that whole approach started off working on it maybe as long as 15 years ago, and they more or less failed at that time. They were using a type of nucleic acid called a PNA, a protein nucleic acid, where the backbone has, well, it has a different structure. And this is a type of nucleic acid that binds very tightly to DNA, to double-stranded DNA, and stops it from being transcribed. But for reasons that they never really worked out, they couldn't quite get it to go. It was too toxic in various ways. I think it was too non-specific. They're working with a different one now, and they report, a graduate student from the same group came to my most recent conference in September in Cambridge and gave a preliminary report which looks as though it's going a lot better. But it was very preliminary. She only really came because I twisted the guy's arm. I think it'll probably be another, well, probably another year before we really see much in the way of detail on that. But yeah, certainly the approach has not been given up on. A large part of the reason why it hasn't been given up on is because it may be a lot more useful in mitochondrial diseases where people are born with an inherited mitochondrial mutation, so all of their cells have the same mutation, so that avoids one of the problems I mentioned. And also they have a much, much higher proportion of their mitochondrial DNA that's affected. So in that sort of scenario, this may be a genuinely useful approach.
Has anyone looked into the idea of just, I know this is a little crazy, but introducing a protein that just damages mitochondrial DNA. Damages mitochondrial membranes to increase the probability that the damaged mitochondria will still get targeted for cleanup.
So,
across the board, across damaging all the membranes.
Right. So your proposal here is that if the mechanism for selective clonal expansion of the mitochondrial mutant mitochondria is along the lines that I've proposed, namely that by their inactivity, they somehow don't damage their own membranes and they stay out of trouble and get broken down less often, then maybe we could actually make normal, non-mutant mitochondria also have low membrane potential and generally damage themselves less often. Is that the basic? That's right, that's what I'm saying, yes. Poke holes in everybody, so that you poke holes in the non-mutant and the mutant mitochondria, so that basically there's less difference between them. Now, that's an intriguing idea. The problem, of course, is that we need a respectable proportion of our mitochondria to be functioning, or we won't make enough ATP, and that's the problem in the first place, of course. So if we do that, then we might reduce the selective advantage, but at a rather profound cost, and we probably wouldn't reduce the selective advantage very much either, because remember, if let's suppose that you've got a factor of 2 difference between the mutant mitochondrial self-damage rate and the non-mutant mitochondrial self-damage rate. What matters is how often a particular mitochondrion is going to be broken down, targeted for destruction. And that will simply be whether it gets there first. So if you reduce that ratio of rates to, let's say, 1.3 or 1.2, it'll still mean that the non-mutant mitochondria get predominantly broken down first, and the mutant mitochondria only have to survive long enough to be replicated, and therefore for the damage to their membranes to be diluted away. So all you'd really be doing in your proposal is making them last long enough by a smaller margin, so to speak. You probably wouldn't change the probability of destruction very much. Any more? All right, thank you very much.